on.
Hi guys, this is the first video for this unit, and it's looking at what is the evidence of hominin evolution. Okay, so what we're going to cover is the evidence, which is for us as fossils, and the most common one we find are things fossilized, so bones, skulls, skeletons. Tools is another common one we find as well. We're also going to look at the dating methods. Yes, that's my cat there. Wondering what that is moving. The dating methods, so things like radiocarbon dating, and a very quick look at DNA analysis and mitochondrial DNA, because we will go over that a lot later on as well. So I just want to go over a couple of definitions, ones that kind of do get thrown around and kind of used wrong sometimes. I admit I probably will use them wrong when I'm talking as well. The first one is hominoids. And hominoids is the word that we use to describe the superfamily, which includes apes and humans, living species, so gorillas, chimpanzees, us, any kind of monkey you can think of, but also ancestral species, so the ones that we're going to look at in a couple of weeks when we start looking at things like Homo erectus, Homo neanderthals, those kind of things. Hominins is the word that we'll probably use the most, and this is kind of where we're not including things like gorillas and other monkeys, but we're talking about direct ancestors of humans. And this is when we split from all the other hominoids about 6 to 7 million years ago. So hominins is our main focus. So when we're talking about hominins, we're talking about us, Neanderthals, any other species that we're going to talk about. Okay, chimpanzees, I guess, does come in there because we did split from chimpanzees between 6 to 7, sometimes you see even more than 7 million years ago. So hominins is the word we're going to use the most when we're talking about fossils, because they're the direct ancestors of us. Oh, it's gone too fast, sorry. So what is a fossil? So most organisms do not actually become a fossil when they die. They have to kind of be in the right place at the right time, I guess is a weird way to put it. But they need to be in very certain specific environmental locations to actually become a fossil. So soft tissues, so things like the brain, muscles, tissue, anything that makes up the body besides bones, basically, so organs as well, can form casts, fossils, or impressions. The skull, teeth, and bones are most likely to become fossilized, but they need to become quickly buried in mud, marine or river sediments. So like I said, they need to die at very specific places, so a river or in the water, and quickly sink. Or another one, covered in volcanic ash. So think about places like Pompeii. You can go there and see the fossils of people because they were covered by volcanic ash, and they've left a cast of that person's soft tissue. So dying in these locations protects them from scavengers and allows for entry of minerals required for fossilization. So what the bone that was once there is no longer there anymore. It's now been replaced by other minerals to be fossilized. Another word you'll hear is I've forgotten the word now. Petrified, and that means it's turned to stone. So here's a quick example. So molds and casts. So a mold fossil is like an imprint. So hard parts of an organism are buried in sediments such as sand, silt, or clay. The hard parts completely dissolve over time, leaving behind a hollow area with the organism's shape. A cast forms as a result of a mold. So you get the mold first, and then water with dissolved minerals and sediment fill the mold's empty spaces. Minerals and sediments are left in the mold like a cast, and the cast is the opposite of its mold. So how a fossil is formed. This is just one example, obviously. So first one is sediment. So an animal is buried by sediment, such as, like we said, volcanic ash or silt because they've died near a river or a lake. Its bones are protected from rotting by the layer of sediment. So as well as protecting the bones, they're stopping any scavengers from getting to it. So then you get layers building up on top of it. More sediment accumulates above the remains. Minerals such as silica slowly replace the calcium phosphate in the bones. So that the bones will no longer be bone, they will eventually be rock. So movement of tectonic plates or giant rock slabs that make up the Earth's surface lift up the sediment and pushes the fossil closer to the surface. Erosion will also do this as well. So say the river's not there anymore and you have rain, it'll wear away at the remaining rock layers, eventually eroding the fossil. Or sometimes you'll also get people having a bit of a guess and thinking, I think there's a fossil here. They'll dig down and find it. So hominin fossils. Because fossilization of human ancestors happens rarely, the hominin fossil record is incomplete. We have bits and pieces of all sorts of different species. We don't necessarily have full fossils. There are a few, but most of them tend to be missing a little bit. So maybe that first step of having the whole individual covered to stop scavengers getting to it hasn't really worked. Or maybe because of the way that the fossil has been lifted up through earthquakes or the movement of the tectonic plates, that the whole fossil hasn't been lifted up. Fossil skulls and teeth are the most common find. The structure of each fossil is compared with other fossils, and similarities indicate how closely related the two are. So when we go to talk about different hominin species, we will look at some of the examples we've got. Oh, sorry, I'm pressing buttons. Will say that we've got some species where people argue that no, actually that should be part of this species. It's always changing, especially as we get new fossils being found. So the dating methods we're going to look at. Radiometric. These are based on the rate at which a certain radioactive isotope decays. So yes, a little bit of chemistry, but I've made it as simple as possible. So isotopes of an element are atoms that have differing numbers of neutrons in their nuclei. So here's an example of an isotope, and the most common one is carbon. So you hear about carbon dating. So carbon-12, that's the normal carbon that we find, has 6 neutrons and 6 protons. Carbon-14 has some extra neutrons in there. It's got 2 extra, so 8 neutrons and 6 protons. So this makes it heavier. So there's carbon-12, it's our stable one, and there's our unstable radioactive carbon-14. So carbon dating. Radioactive carbon-14 joins with oxygen to make carbon dioxide and becomes incorporated in plants by photosynthesis. The ratio of carbon-14 in the atmosphere and then into us is only small, so it's about 0.1%. So we eat the plants, and our tissues then contain the same ratio of carbon-14 to carbon-12. So we actually eat this radioactive carbon-14, and it's then becomes in our tissues just the same as our carbon-14 is. Once the organism dies, it no longer takes in any new carbon atoms. We're not breathing anymore, so we're not producing any carbon dioxide in our mitochondria from the reactions that occur in there, because we're dead. We're not eating anymore either. So as the carbon-14 decays to nitrogen-14, the ratio can be used as a clock to date how long it has been since the organism died. However, this is where carbon dating kind of isn't as good as we'd like it to be. We can only date samples less than 60,000 years old. So things like, if we talk about Lucy, who is Australopithecus afarensis, one of the most famous hominin fossils, she's kind of maybe 4 million years old. So carbon dating is no help to us there. This graph really helped me understand how it all works. So carbon-14 has a half-life of 5,730 years. So in 5,730 years, half of your carbon-14 atoms in your body would have turned into nitrogen-14. Okay, let me bring up so I can draw on here. So, so after one half-life, you're only going to hit, so that's after one half-life. Three half-lives, you've only got that much left. Then after 57,000 years, less than 0.1% of the original carbon-14 is still present. Move the cat out of the way. So it's the radiation being given off. There we go. The beta particle there that is actually detected. So this is a little picture. Obviously, the first thing you should see about this diagram being incorrect is that we're talking about dinosaurs here. They died out, 60 million years ago? So carbon dating isn't actually going to work, but it's a good example. So if we start from the carbon-14 atom, joining with the oxygen molecule, we then get carbon dioxide molecule. It's taken up by plants. This guy here is then eating it. Then he dies for whatever reason. At his death, he has 16 atoms of carbon-14 in him. After the half-life of 5,730 years, there's going to be 8 atoms of carbon-14 left. After over 11,000 years, there'll be 4 atoms. After 17,190 years, you've got 2 carbons left. So every 5,730 years, you halve the number of atoms of carbon-14 that are there. What I'd like you to do is go back and watch some of these videos and animations. First one is the radioactive carbon animation, which will give you a little bit more information on how it works. And then there's one there about becoming a fossil. So it shows the special conditions that are needed to make a fossil, because not everything that dies makes a fossil. So, as we said, carbon dating only works up to, say, 60,000 years. After that, you're not going to be able to get the results. So potassium argon dating is used for things that have probably been dead much longer. So potassium 40 decays into argon 40 and calcium 40 at a known rate. Okay, the half-life for potassium 40 is approximately 1.25 billion years. It's the rock that is around the fossil that is tested rather than the actual fossil, because the fossil has changed slightly differently. It's the rock around it that we test. So every time it halves, okay? So as potassium 40 decreases, the percentage of argon 40 is going to increase. It sounds a little bit tricky to understand, but it can be used for things that are much older. We're talking millions of years old, billions of years old here. Accuracy. So potassium argon dating is not as accurate as carbon dating, but we can use it for the really old stuff. So potassium argon dating has an error margin of plus or minus 0.25 million years on a sample that's 2 million years old. Not too bad. Whereas carbon dating has an error of plus or minus 40 years. But remember, carbon dating we can only use up to something that is, say, just over 60,000 years old. So one of the new ones scientists have been using is DNA analysis. And there's been quite a few New Zealanders involved in this overseas, and we're going to talk about them a little bit later on in the unit. But DNA analysis is kind of the new one that we use. So the relatedness of species can be discovered from the differences in their DNA. One of the ways we do this is with DNA to DNA hybridization. So this is a way to compare the genomes of different species by measuring how genetically similar two DNA sequences are. This is DNA to DNA hybridization is one of the ways we found out how related we are to chimpanzees, and we're sitting at kind of like 98.2% similar. And saying that, I think we're kind of like 90-something% similar to a banana as well. So the more related species are, the fewer differences between their genomes there are than distantly related species. So hybridization has shown, there we go, that chimpanzees are our closest living relatives with over, 98.6% of their DNA sequences are the same as ours. Gibbons, which we would have split off from, I think, maybe 10 million years ago, we're only 94.7% similar with our DNA to them. Rhesus monkeys, which we then split off from maybe 10 to 12 million years ago, is 91.1% similar. So the closer we are from splitting off from a species, the closer our DNA is. Here we go. So humans and chimpanzees, we split off from each other somewhere between kind of 6 and 7 million years ago. Gorillas and whatever our ancestor was there, split, say, 8. Gibbons, a bit longer, as we said before. So mitochondrial DNA, this is a really important one, and one that is really being focused on at the moment. So mitochondrial DNA, and especially we've got, again, some New Zealanders, scientists who are really interested in looking at mitochondrial DNA. This has been used to investigate human origins and when humans began living in certain parts of the world. So mitochondria are only passed from the mother to the child in the cytoplasm of the eggs. So your mitochondrial DNA is passed down your maternal line. You don't get any mitochondria from your father. The mitochondria are found in the sperm, but in the tail of the sperm, and they're not put into the egg. Only the DNA from your father is put in. So mitochondrial DNA can act as a molecular clock, giving an idea of how long ago members of a species had a common ancestor. So here's a little bit of an example. So there are mitochondria in a sperm, but it is only the DNA that's actually put into the egg. So mitochondrial DNA can be traced back through your maternal line. So an analysis of mitochondrial DNA found that all humans probably had a common ancestor in Africa somewhere around 150 to 170,000 years ago. So skulls of modern humans of this age were found in Africa in 2003. Mitochondrial DNA is used as evidence for the out of Africa theory that we're going to look at in a few weeks, which talks about when humans moved out of Africa and started living in other parts of the world. So that, I'm pretty sure, is our last slide. So I hope this video has given you a bit of an idea of what the evidence is for human evolution and how things are dated. From here, you've got a few activities, which you'll find in the Google Drive folder. Remember, if you have any questions, please let me know. Thank you.